ligand molecular docking. The protein ligand molecular docking is the technique which is widely used in chemoinformatics to analyze the interaction between the protein and the ligand, like we have here. The key and the lock. The key is the ligand and the lock is the protein. There are some binding sites in the protein for the interaction. And we are trying to establish the interaction via different algorithms. So I will not gonna give you the full introduction of the protein ligand molecular docking today, just an overview of this technique. And the person who invented this theory is the German scientist, Professor Fischer, who introduced this paradigm in 19th century. And so far, since then we are using this theory to understand the investigation, protein ligand, to understand this protein ligand interaction. And there are different algorithms, different protocols. Right now, we have the protocols which are very sophisticated, using machine learning, deep learning algorithms like DeepDoc, GNINA algorithms. The old algorithms are based on the genetic algorithm. You also have Monte Carlo. You also have some kind of simulated annealing algorithm. Sometimes you can do this manually, like a visual molecular docking. When you take the ligand and trying to put this ligand in a protein ligand binding site using some haptic device. This can be also implemented in the virtual reality environment. So we have the virtual reality environment. We can actually try to create something like that to simulate these processes, to simulate this interaction in a virtual reality. So this would be also a very interesting project. And genetic algorithms are right now so very widely used, like Autodoc Winner, SMINA. We have Glide, we have Malegro, we have Moya genetic, we have Moya docking algorithm, we have Dymodoc, which was designed by me and based on the genetic algorithm as well. We have HatDoc, we have Doc 6. I think this is the 6th version. So there are different algorithms, different software programs. So one could use the right algorithm for the particular project. And we are using the new algorithms and the old algorithms, trying to compare, trying to do the benchmarking. Some algorithms are more powerful than the others, and using different scoring function. And usually when you do the molecular protein ligand interaction, you have molecular, some kind of a scoring function. You have to score the molecules in order to understand in order to find the best binding pose. Once the molecule is rotating, it generates a lot of different conformations within the protein. So these conformations can be analyzed by using some scoring function and ranking. So in order to do the ranking, you have to produce some kind of a score. These scores can be different. They can be free energy based scores, like biochemical, like a physics derived scores, or they can be empirical, they can be hybrid. Some people devising the scores using some statistics analysis. Right now we have also machine learning scores. Some people even do some kind of data mining, like using the artificial intelligence to post-process the IC50 or the experimental binding constant. So far, I'm not sure whether it's really represents the right scores to do the ranking, but they're trying. So, but not that many papers published so far who are trying to implement a similar methodology. The score is the representation of the free energy. The free energy is calculated by using the Gibbs free energy of binding equation, Gibbs equation, to calculate the Gibbs free energy of binding. And it's just a sum of different the energy binding terms, like electrostatic, electrostatic van der Waals energies, rotational energy, translational energy, bond energy, salvation and desalvation energy, and stuff like that. So when you get the sum, the energy sum, like the total energy, it will represent the binding energy. You can also calculate the binding energy by calculating different and the potential energy of different substituents used in a molecular docking, like the target, ligand, and a complex. And, usually, you don't really have a solvent in a molecular docking technique. Therefore, you have to use the external programs like fast DHR algorithms to analyze the salvation, the binding energy based on the salvation models. Usually, there are two main models to investigate the salvation. Is the MMPBSA and MMGBSA models. It's Boson-Boltzmann molecular mechanics, Boson-Boltzmann surface area in generalized Bohr surface area. And by implementing this function, you can also calculate the binding energy based on the salvation, implicit salvation models. So this technique is is widely used in drug design, in the material science. In the material science we are trying to also implement this technique in the material science, but most of the time people are using it in a rational drug design when you get a relatively huge library, so you can screen the library, and produce some heat molecules. Then you will take the molecule for the heat to lead optimization again, do the cycle, then again you get some heat molecule, or until the heat molecule is very well optimized, then you can proceed with some other studies like in vitro study, or you can implement different in silico techniques like molecular dynamics. So the protein ligand is a very rough estimation of the binding, because you don't really have a solvent, so you're neglecting different important parameters. The environment is a kind of a gas environment, like a vacuum, so you don't really have a interaction in vacuum, so it's very artificial. Therefore, you cannot really rely on the molecular docking. You can just approximate the interaction based on this conformational sampling. So people have to always have to pay attention that this technique is the only very approximate evaluation of the binding. So one could and should use different, more sophisticated algorithms like molecular dynamics when you actually have the water molecules and the system is moving, so you can have a more you can mimic actually the environment which takes place in the real life. Okay, so what kind of projects we have right now? So this is the overview of this project. I will talk about them in the next lectures. What kind of algorithms we already designed, and what are the molecules we are using in our projects, and what is the idea behind it. So this and using molecular docking techniques. So the first projects I would like to talk about is the project related to the COVID-19 pandemic, and the idea to generate some substance in order to treat to inhibit the replication of the virus within the cell. And here we have the project where we think that we can inhibit some viral enzymes like polymerase here to stop the replication of the virus. The other pathway to inhibit is the binding of the virus to the cellular membrane, and this binding will be mediated by the cellular receptors like TMPRSS2 and ACE2, and the spike protein of the virus. So The idea was to generate some drug to bind to these receptors, to occupy the protein ligand binding sites, and the interaction of these proteins and the protein-protein interaction is not possible anymore. As you can see here, we have some drug. We call it chemostate substance. It has some lipophilic centers, which are probably involved in an interaction with the lipophilic binding sites within the receptor within the spike protein of the virus. So they think that if we inhibit the spike protein, there will be no interaction with the cellular receptors anymore. This is the co-receptor, this is the main receptor, ACE2, where the spike protein is in a contact with. So we're also trying to do some kind of a drug repurposing. So we have some antiviral substances like beclabovir and remdesivir, and we were trying to analyze the interaction of the drug with the protease. So here's the protein ligand interaction, and as you can see, the substances are in a catalytic site. So this might be the mechanism of the inhibition of this virus. And this is again the study protocol, what we have to do, what we could do to generate some lead substances. We could scavenge the substances using different databases. Some natural compounds can be used as well, like Afro database and non-PDB natural compounds database. Then we can come up with some thousands of molecules with different molecular weight. We were trying to use machine learning techniques or just the standard algorithms. We can also enrich the pool with some FDA-approved antivirals to increase the amount of the compounds. So we get more. Now we have more substances, close to 2,500 substances. And then we will do molecular docking with the main protease of the virus. And finally, we will analyze the compounds using different algorithms to predict biological activity, NMD simulation. So we would come up with 6 or so potential leads. So this protocol also can be used to, this is the published protocol. So this protocol can be used in our project. Now we are trying to optimize the protocol, and we are also thinking that it is important, if we have the substance, to improve the solubility in order to dissolve the substance in the water, because sometimes the substances are not soluble, therefore you have to use very aggressive organic solvents like DMSO in a high concentration. Usually you don't have to increase the concentration up to 10%, because this would also be very harmful for the cell population, and the cells did not survive due to the very high concentration of the DMSO in the medium. And the idea was to do the formulation, and the formulation is the complexation of the guest molecule. As you can see here, the guest molecule is the ligand, and the host molecule is the cyclodextrin. As you can see here, the cyclodextrin is the oligosaccharide made by the glycosidic residues with a number of hydroxyl groups. These hydroxyl groups can be substituted with different moieties, so we can actually make the cyclodextrin more lipophilic, less lipophilic, changing its geometry. So the molecule could be also optimized in terms of the solubility. As you can see here, if we will substitute the hydroxyl group for the hydroxypropyl beta residue, so the substance will be more lipophilic, and not very lipophilic, but still there is a change in the lipophilicity. Sometimes we can make also substitution for the methylate. We will methylate the cyclodextrin, so we will produce methyl beta cyclodextrin. We can also add some inorganic moieties, although this is not very common. The formulated molecule so far is the beta cyclodextrin, because it has a bigger binding site, like the cavity. It has the bigger cavity than alpha cyclodextrin, and 7 subunits. In comparison to the alpha cyclodextrin with the 6 subunits, so cavities is smaller, and probably it's not enough to do the efficient formulation. The other cyclodextrin is the gamma cyclodextrin. Gamma cyclodextrin has the 8 subunit, and also is not very common because the cavities is bigger and make it loose for the ligand to be efficiently formulated, and the complex probably would lack the stability. The well-characterized cyclodextrins contains 32 anhydroglucopyranose units, and one could also synthesize the cyclodextrins, but they are not really widely used in the pharmaceutical industry. As I already mentioned, the cyclodextrins are widely used in drug delivery. So in Russia, there is a market in Russia, but mostly in Europe and in the US. And the gamma cyclodextrin, the beta, alpha, beta, and gamma cyclodextrins are usually largely recognized as safe by the FDA. So they are very promising excipient molecules to do the formulation with different hormones, with different anesthetics. In our projects, we use different anesthetics like propofol, propofol substances, the general anesthetics for the widely used in anesthesia and clinic. And we were successful to improve the solubility of the substances. So therefore, cyclodextrins are very promising. On the other hand, cyclodextrins confers solubility and stability of these drugs as well. They might also increase the stability if the drugs are not really very stable. They are controlling the degradation of these complexes and the mechanism of this protection is probably that the substance is protected from the pH and this might increase the shelf life of the molecules. Additionally, cyclodextrins are also shown to enhance mucosal penetration of the drug. We've analyzed the drugs on the ability to permeate the blood-brain barrier, but it's not really clear how the cyclodextrins are involved in this process. Either they will transfer the ligand to the site of action, to the blood-brain barrier interface, and then dissociate. The complex dissociates and the drug is released from the cyclodextrin cavity and permeates through the blood-brain barrier, and the cyclodextrin remains in the bloodstream. Or maybe there is some other mechanism on how the cyclodextrins might interfere with the permeation of the drugs. Probably the cyclodextrins are interfering with the lipid membranes, and this is the fact, which also raise a concern on the toxicity of cyclodextrins, because it is known that cyclodextrins would also deplete the cholesterol molecules from the lipid membranes. This might be also dangerous in terms of the toxicity. Once the cholesterol molecules depleted, the cell functions are impaired. It would also have a negative effect on the lipid rafts and some proteins associated with the lipid rafts, some transporters, very important transporters, and it would induce the toxicity for the cell. So one could also could use the cyclodextrin with extreme care, not to enhance the toxicity of the cellular toxicity by using it. Okay, so we can have a break, and we will start in 15 minutes.
Yes, sorry, and if we get questions, yeah, we're gonna ask those in the end of the lecture.
Yeah, no, you can just ask the question. Sure.
Okay. Okay. So I'll start with the one on that slide. Could you please explain a bit the isotherms? pictured on the left.
Yeah, the other terms I will explain when we will go back to the lecture in 15 minutes.
Okay. The second one was about the first slide, but it's rather short. May I ask you to?
Yeah,
Or maybe yeah. After the break, I'll ask those.
yes,
Okay.
after the break, I will explain it to you.
Oh yeah. Okay. Sorry. Thanks.
Okay, let's continue with the lecture, and I will explain the results we have. We have a collaboration with our partners in Germany, in Würzburg. They have different cyclodextrins to formulate with. These two cyclodextrins were chosen to be the good candidates for the formulation. One is the sulfa-butyl cyclodextrin, and the other is hydroxypropyl cyclodextrin. And we wanted to understand the binding of the cyclodextrins at different pH levels using the same ligand as a promising candidate to inhibit the COVID virus receptors, like Spark, like this one, spike protein. And the solubility isotopes. The solubility isotopes are the curves or phase diagrams, and they depict the relationship between the solubility of the substance and the temperature and pressure under specific conditions. And these diagrams are widely used in medicinal chemistry, chemical engineering. They can be temperature dependent, pressure dependent, mixed solubility, binary phase diagrams. And in our experiments, we used the standard procedure to produce the solubility isotopes. They were using HPLC technique, and as you can see here, we have different results at different pH. We measure the concentration of the substance after by adding different concentration of the cyclodextrin molecule. So the idea is when we add in the concentration, more concentration in the cyclodextrin is the weight percent. So after some time, we are measuring the concentration of chemostat, and if we have higher concentration of cyclodextrins, we also have a higher concentration of chemostat in our solution. So when we compare these two solubility isotopes, we can see that after this, at the same concentration, cyclodextrin concentration, we have higher concentration of chemostat ligand in a solution, which means that the cyclodextrin is bound more tightly to the cyclodextrin in comparison to the first formulation. Here we have the same concentration of the cyclodextrin and the high concentration of the free chemostat molecule. That means simply at the same concentration we have more, which means that the binding is probably less is less strong in comparison to the hydroxypropyl beta cyclodextrin. So from these isotopes, this also holds true for this old solubility isotopes. If you look at the high pH isotopes, they're quite close. At the physiological pH, the chemostat concentration is close to 40 milligram per ml, and for sulfabutyl beta cyclodextrin is close to 50. So we have higher concentration, which means that more ligand is present in the first system. And at low pH, we have very different isotopes. So the concentration, also the effect is present here, that if you decrease the pH, you also decrease in the binding of the host guest, of the guest binding to the host. So once the pH is lower, then the concentration is going up in comparison to the lower pH. So this is the experimental result from the solubility isotopes. One could approximate the binding affinity. Here's just a simple information that the one cyclodextrin is the better than the other in terms of the binding. So we can also approximate it using the molecular docking technique by modeling the host-gas interaction. But the problem is that the cyclodextrins are complex molecules. They have different, they have a very, they have multiple groups, like hydroxyl groups, and these hydroxyl groups, they are substituted for hydroxypropyl, and we know that the degree of the substitution is 4 for our cyclodextrin molecule. And this would also create different conformers, different conformer molecules, as written here, because the degree of the substitution is random. So it's of course more than 4 molecules, but we substituted only the big mouth of the cyclodextrin. You got a smaller mouth of the cyclodextrin here, which represents, if you have totally 21 groups, here you have, in the big mouth, you have 7 groups, and in a small mouth, you have 14 hydroxyl groups, which makes the molecule very hard to model. For these reasons, we just substituted only the big mouth of the cyclodextrin using the randomization algorithm. So we just substituted for groups in the 7 group diameter. And we came up with this isomeric structures. And in case of the hydroxypropyl molecule, we got the 7 groups, the degree of substitution is 7. So all the hydroxyl groups in the molecules I substituted. So you would always have also only have one structure, which is easy to model. After that, you could do the optimization, like some kind of conformation search to figure it out. What is the dominating structure? We have to come up with the structure which is more prevalent in the solution. So one could simply run this in a dynamics as a molecular dynamics in the solution, and the large cluster and the conformation, which represents the large cluster, would be considered as a dominating conformation for the modeling. So this was not done so far for our cyclodextrins, but it should be done to come up with this dominating conformation. And once we have a dominating conformation, we can just simply do the host gas interaction with our target molecule. The same could be also performed for the cyclodextrin beta hydroxypropyl sulfur butyl ether cyclodextrin. So as you can see, the project is not really very simple. We have to do a lot of optimization, simulation, conformational search, optimization of the cyclodextrin molecule, the free energy extraction, in order to compare with the model with the experimental data. The cyclodextrin, is quite appreciated molecule to do the modeling, because it has a very, very good binding site, like shown here. When you have the formation of the complex, it's usually quite stable in the molecular Dawkins studies. We also analyzed the ligand in terms of the druggability of this molecule. So we produced we analyzed the substance for different rules, such as Lipinski's rules, to find whether we could find some violations. And we actually found that there are some violations. But on the other hand, the druggability is not really very, very bad in terms of the standard druggability. So you can see that the red is kind of the standard druggability, and the substance, the rose map is quite close to what we have here. So I mean, the substance, if we would like to proceed with the substance, maybe some modification also might be important to improve the druggability of this compound. The TPSA is a bit high. The log P score is quite acceptable for this ligand. One could also use the lower log P. So for this reason, we would use the cyclodextrin to decrease this lipophilic number of this substance. But again, one could also modify the molecule to make it lower, to make it soluble, or to use some other molecules for the formulation. The project is still ongoing. We were able to generate these conformers. Now we are running the dynamics to extract the representative conformation of this molecule. The other project is to analyze the different proteins. I would just give you some introduction what we have here. Actually, we have some results. But maybe we can just proceed with some other lecture. to skip this experimental data for the next lectures, and I would just probably give you the introduction on the molecular docking, because we already started with the molecular docking. I already specified what kind of projects we have. Maybe we can just go in more details on what the techniques can be used in particular, why the molecular docking, what are the fields which you can use the molecular docking, and so on. So the molecular docking, as I already mentioned, is a universal procedure which is used in the chemoinformatics, in the rational drug design. It is very important to come up with the right structures for the further analysis, and one could use the single molecular docking or multiple molecular docking, like the virtual screening here, to identify the compounds modulating disease-related targets and their optimization. You can also use ligand binding rationalization to identify the structural determinants necessary for the efficient receptor binding. You can also predict the adverse drug reaction. The adverse drug reaction meaning that you could analyze these proteins which are affected by the drug, like housekeeping proteins, and if they are affected, there might be the adverse effect and the negative and the toxic effect on the cells. So molecular docking might be used in polypharmacology, identification, optimization of compounds that simultaneously modulate a set of targets involved in the same disease. So like the competition between all these compounds, and you can structure the substances to have an idea how they are competing for this particular receptor. And some drugs are promiscuous. They tend to bind various protein targets, and also this might be due to the different side effects associated and the adverse reactions associated with these particular drugs. Sometimes molecular docking can be used for the drug repurposing, and we also had some projects. I will talk about it in the next lectures, where we can identify the novel therapeutic relevant targets for already marketed drugs and known chemical and natural entities. When we have the database of the FDA approved drugs, so we can screen them in terms of the unknown targets to identify the best bioactive substances. So molecular docking technique is quite ubiquitous. You would probably use it in every experiment possible where the molecular interaction is taking place. Usually the chemistry is about the molecular interaction and the molecular design, and when you come up with the new materials, you would like to know how these materials would interact with other materials, and how to, if you're creating the new biologics, let's say antibodies, you would like to know how the antibodies and antigen are formed, and for this you could use the molecular docking. There are different docking techniques in terms of the in terms of the molecules which are used for the interaction, like protein ligand or protein can be morph ligand or some formulated molecules like host gas interactions, host gas system. This can be also performed on some surfaces like hydroxyapatite on a crystalline structures as well. But of course, this is less precise because of the right now we are trying to implement this algorithm, which are very well known for the protein ligand interactions. So sometimes they're not very precise when we are changing the system. Like hydroxyapatite surfaces and some antibiotics. Right now, there is a lot of research going on to improve the predictability of the molecular docking algorithms using artificial intelligence and different statistical methods. I already mentioned diffdoc, which is based on the reverse diffusion algorithm. In the molecular docking, you have the pre-docking screening when you set the selection of protein conformers for virtual screening, and then improvement of the scoring function. And you can also do the post-docking screening, like ad hoc. The other term is ad hoc screening, when you can implement the salvation models like MMPBSA, GBSA models to calculate, to simulate the environment, the aqueous environment. You could also do some kind of post-docking screening in a pose rescoring. Sometimes pose rescoring is also very important when you get different poses, so you can rescore them using some chemoinformatics techniques like topology, like the molecular topology, or you can use some geometrical representation. We did this using the symmetry, using Voronoi algorithm to associate the symmetry with the rotation of the ligand. And of course, this would also affect the binding to the receptor. And you could also add additional techniques to the molecular docking, like molecular dynamics, where you could identify the representative conformations for molecular screening, as I already mentioned before, using the cyclodextrin molecules. And here, actually, we are doing exactly the same thing. We're trying to do the search of the representative conformations, and after that we will do the molecular docking. Kind of first we do the dynamics and then the molecular docking. But of course, in most of the protocols, people are starting with the molecular docking, and then which is less time consuming, less precise, very approximate. And then when you get the small number of ligands, you can confirm this information by using the molecular dynamics. It is also possible to investigate the stability of the complex using, for instance, Monte Carlo techniques. So you will do the sampling of the molecular docking results and so on. So molecular docking is the docking technique, is the computational technique to determine the binding activity between the receptor and the ligand. The ligand usually is a drug-like molecule, not very big. Usually it contains up to 70 atoms, sometimes more, sometimes less, and the rotation, the suitable for the rotational effect to within the protein ligand binding site. So the molecular docking tries to find the best matching. So once the ligand is rotating, it tries to find the best matching between these two molecules based on the appropriate scoring function. And the molecular docking methods usually are optimized when the receptor is rigid and the molecule is flexible. Therefore, this is also the limitation of this technique, because you have the kind of a preserved conformation of the protein, so you don't know whether this is the open conformation or its closed conformation. The binding site is also kind of rigid; it's not moving, and usually in reality, it's not the case. So it's very mechanistic, very approximate, and the water is also not present, and the water is the conduit sometimes between the ligand and the receptor, participating in the binding. Also, there are some ions which are very important for the binding. The pH is very important. You already saw, right, that the pH would interfere with the binding, with the protein ligand or the host gas interaction. The charge is different, so pH is different, the charge of the molecules is different, and the binding is different. So here we don't really have any of these parameters. We just have the binding site and the idea is to make the ligand rotate to provide the best matching between these two molecules. So very, very approximate. Therefore, sometimes it's also very promising to generate a lot of conformers, the protein conformers, and screen them all in terms of the binding. But this is very laborious, and so people don't really do it. They rely on some experimentally derived data from the PDB, the protein data bank. Once you get the protein ligand complex co-crystallized, so you will just assume that this is the binding conformation. This is the most relevant structure for the ligand to bind, and you just do the modeling. So this is, of course, the best model you can get in terms of the experiment and accuracy. If you would try to do the homology model, usually sometimes it's not really possible to understand the interaction between the protein ligand, and we did similar experiments using some taste receptors when we were trying to analyze protein ligand binding sites. In one experiment, we used TAS1R3 receptor, which is responsible for the taste, for the interaction with the different taste molecules like umami, and the domains were different in terms of they were very complex. The binding domain, the ligand binding domain was called the Venus flytrap, and the morphology and the topology of this domain was very complex. It was kind of a bulb, extra. There is a transmembrane domain, and this is the extracellular domain. And this extracellular domain is important for to sense the taste ligands. So when we tried to analyze, we couldn't really produce the meaningful information, because as we assumed, this domain was not really open. So you have to do some optimization of this domain, see how it behaves in a water-like environment, to see the dynamics of this domain. So we were not able, and the binding site was too narrow probably for the ligand to squeeze in. So we were not successful. So this is the big drawback of the molecular docking, if the receptor is rigid. Of course, some algorithms are able to add some flexibility to the receptors. Usually, you can specify small number of amino acids, so you can make them also flexible. They would rotate, but then again, this is not really fully flexible protein structure. So this would also be probably the conditions are closer to the reality, but not as in molecular dynamic simulations. But the ligand is flexible if it's relatively small, only 32 torsional bonds are flexible, and if we have more than 32 torsional bonds, then the algorithm will not really consider these ligands for the simulation. So you have to either improve the ligand by decreasing the amount of the torsional bonds, or you have to compile the software by increasing its number by probably 62 or so, which is not really parameterized for the standard molecular docking algorithms. Various genetic algorithms are widely implemented in the molecular docking methods. These algorithms described ligand conformational changes by mimicking the process of evolution, mutations, selections, and crossover. This is a genetic algorithm. There are different, of course, algorithms, but it's mostly prevalent. So what are the molecular docking algorithms or software? Well, one could say that they're not algorithms, but rather they're programs designed by different groups. The first algorithm which is widely used so far is the Autodoc, and the Autodoc Vina. It's the derivative of the Autodoc algorithm, which you could run on multiple CPUs quite fast, and it's actually very useful for the virtual screening. The first one is more like for the molecular docking, and the second one is more suitable for the virtual screening. These programs were designed in the US, in the Scripps Research Institute in Olson's laboratory in 90s, and in 2010 they designed this Autodoc Vina, still improving all these molecular docking techniques. You also have some very sophisticated Autodoc tools where you can process the ligands, analyze the binding sites, and so on. So they're working together, actually. You can prepare the input files and send it to the Autodoc program. They are based on this one is based on the genetic algorithm. This one is based on the global optimization technique, some kind of a hybrid technique. So they are actually quite good in terms of the accuracy, if the system is not too complex, and if the validation is provided. Sometimes they're not sure. There is a big dependence on the binding sites. If you have you have to process also the protein structure. If the protein structure is not processed, the algorithm is not working, and the accuracy is not good. So it's a bit tricky. It's a bit tricky methodology, but still widely used in chemoinformatics projects. And you have the physics-based binding energy, which is called the Gibbs free energy of binding, RT/Ki, where you can also devise the Ki value, the inhibition constant, and from this inhibition constant, you would have some idea. At least you could try to correlate it to the experimental inhibition constant, or sometimes the IC50 values. So it's a very big benefit of this molecule, this method. The Voronoi tessellation is kind of a binding site, the algorithm to determine the binding sites. And there are some also hidden Markov models algorithms, but they're not very widely used. And there are new algorithms, the regression algorithms, and the algorithms, the diffusion algorithms. And the regression and the diffusion algorithms, they're relatively new algorithms, just published and developed and published a couple of years ago, especially the diffusion algorithm, where you are using the machine learning approaches. And one of these algorithms is called the DiffDock, where you are using this reverse diffusion protocol to do the molecular docking. Was developed by the MIT, and right now is claimed to be the most accurate algorithm in terms of the clustering. So this scheme tries to provide the overview on different algorithms, like what kind of algorithms we have, and we have global feed, systemic, some kind of a systemic algorithms, trial and error like protocols, where we can use genetic algorithms, simulation annealing algorithms, taboo search. And I actually didn't use this algorithm, taboo search, so I cannot comment on it. But the genetic algorithm and the Monte Carlo simulated annealing, Monte Carlo simulated annealing, the other quite widely used algorithm. You also have a systemic post-generation, like a stochastic optimization algorithm, which is GLIDE, incremental construction, the tree search, the Hamadoc, the tree search is used in the Hammerhead and FLAG6 algorithm. Some algorithms, the graph match algorithms used in the DOC3, FRED, E-HITS algorithm. E-HITS program uses multiple copy rigid algorithm. But the main algorithms are the genetic algorithm, the Monte Carlo algorithm. Simulated annealing is also used in the autodoc, so induced fit and the simulated annealing we can use in the autodoc program. So you can also subdivide these algorithms like more random algorithms when they have the random functions, and these algorithms are more directed. There are also some machine learning algorithms that are not present here, but they are also more directed. So kind of you have the information on the pose, you train the model, and then you do the docking, and of course you will very high matches of the poses to the experimental structure. So what is the genetic algorithm? Genetic algorithm is the kind of optimization algorithm used in different fields of science, also in chemoinformatics, in informatics, in computational chemistry, in the industrial-oriented sciences like mechanical engineering and so on. So what is it about? You have some kind of a genetic chromosome, which is a kind of a binary. This represents your ligand, and ligand is kind of encoded in an interaction. They're encoded in this genetic chromosome. Then you just specify the three operators like crossover, mutation, and selection. And according to these operators, you're starting to mutate the sequence, so the artificial digital genetic chromosome to produce different chromosomes. And then you will have some kind of you will integrate this into the scoring function, and according to the scoring function, according to the operators, crossover, mutation, and selection, you can choose what kind of ligands you would have as an output. Like this operator chooses the individuals with the lowest values, and the lowest values are the energies, and then those with the high values are not really chosen for the next iteration, and so on. So you will have various iterations, and you can only choose the individuals with the lowest values according to the fitness function. So you need a fitness function like the scoring function, and then by providing these operators, you will screen the molecule the poses, the rotations, the rotor mass, which with the lowest values of the score, with the lowest values of the energy, and the rest is just discarded, as you can see here. So after that process, after some iteration, you will come up with the best candidate, which has a very low value. That's it. And this is what it's all about, the genetic algorithm. After that, you will get the pores that will give you the probabilities. Most likely, this is high because the probability is high, and the pose is most likely to be the one with the lowest value of the energy, and this might be your candidate for the highest affinity. So this is the idea of the genetic algorithm. The other algorithm is the Monte Carlo algorithm, and here you have some rules, the mathematical rules, and this is the algorithm. You have the configuration of a molecule. Usually it's the energy. It's the energy of the molecule. And then you have a random move of the configuration B, the energy of the B. This is the energy. This is the energy of the protein, and this is the energy of the ligand, because the ligand is the moving part of the system, and the protein is the rigid. And then you have to accept the move when you have the energy differences according to the rules provided here. When the energy of the B state is lower than the A state. So actually, this is the kind of a starting conformation. This is the starting conformation, and then it's not really a receptor. This is the starting conformation of the ligand, and this is the moving conformation. And then you have, because with the starting conformation is a kind of a initial conformation, the energy A, and it means initial. Yeah, the kind of initial in this. This conformation is a moving conformation, something like that. And then you got the differences in these energies. When the energy of the initial conformation low, then the lower than the energy of the moving conformation. Then, or if it's high, then accept the probability, and you will calculate the probability according to this equation. So if the energy is higher, then the energy is than the energy of the moving conformation, then it's good for us, because then we would like to find the energy which is lower, because then when the energy is lower, then the affinity is higher, and vice versa. Therefore, if the energy is high, then the affinity is very low, so we don't want it. We don't want this entity. Then we will provide the rule to concentrate on the energy which is lower than the initial conformation. And this is the A. This is A. The conformation A is higher than the conformation B. Then we are searching for this conformation. If the conformation B is higher than the conformation A, so then we we exclude this pose from the further analysis, and we will calculate the probability based on this equation. That's it. So it's a kind of the probability function is a kind of acceptance function. So to accept or to reject the right pose with the right energy. Actually, the molecular the standard molecular docking is all about the energy. So it's kind of the P is also quite close. This equation is quite close to the Gibbs free energy of binding. The equation of the Gibbs free energy of binding. This is also the function, the exponential function. You also have the KT, and so then you have to calculate this probability. And this is the computer modified script, how you can actually put it in the computer and calculate the energy. When you have initial setup, you're proposing the move, then you evaluating the probability ratio, this one. Then you accept or reject, update atomic position. I would use the update the atomic positions because we don't really use the electrons. So this should be the atomic position, and then calculate the local energy of the pose. Then you can either reject or accept this pose, and then you will have the output results. So this is kind of straightforward algorithm. Then you will get the right pose according to this algorithm. So this is the Monte Carlo. The Metropolis criterion is actually the criterion how to accept and reject the right pose according to the acceptance ratio. Okay, what are the other algorithms? There are some other algorithms called the simulated annealing algorithms, and they are also used in the molecular docking experiments, and you can start with the random run, also positions, orientations, conformations. Then you can make the random state changes with the probability dictated by the temperature. So in the equation, you have a temperature function, and then you will have the uphill random changes. You are elevating the temperature, and then see how it would proceed with the random states of the system, like provided here. So and then you will have the kind of a chance to reach the minimal energy in comparison to the local optimum. So the idea is to find the global optimum. That's why you have this simulated annealing, like a cooling. Then you will have some. So then you do the elevation of the temperature, and then you will slowly reducing the temperature after each move to reach the decrease in the energy, and then you are repeating, you are iterating, and then you terminate. Finally, you terminate the process when the temperature gets very small. Then again, you have the acceptance according to this equation, and it will help you to find the global minimum of the system. So this is actually what is all about the simulated annealing. The simulated annealing is why it's called simulated annealing, because it was found in the metallurgy. It's a probabilistic technique for approximating the global minima by the given function. So it's a kind of mathematical technique, and the name of the algorithm comes from annealing in metallurgy, when you are heating the metal and controlling the cooling of the material, altering its physical properties. So when you're heating the material, then you're cooling, like you saw in some shops, to create in some metal tools, like blacksmith shops. And they are heating it's called the tempered steel. Yeah, when you temper steel, you will make it very hot. So they also add some chemicals to decrease or increase the content of carbon in the metal. So I mean, if you're interested, you can just go through the metallurgy and how it's done. So this was actually taken from the process to simulate this process was actually to simulate this process in the metallurgy to come up to the global optimum of the system. And this algorithm is also presented here in a more concise way. When you do the generation, when you judge the transition, do the cooling, you of course have a temperature elevation, and you will have the global optimum of the system. That's it. So there are also some scoring functions to rank the right molecules, and you can also subdivide the scoring functions in different classes, like shown here. The first class is the force field-based functionals, where you can use the force field estimating the affinity by summing some energy of the protein ligand interaction, like van der Waals and electrostatic interactions, putting all together in a kind of a force field. And by doing this, one could also calculate the interaction of the unknown ligand. The other is the knowledge-based, where you can have a statistical observation and also create some kind of statistical potentials. This can be also taken from the literature. So for instance, some statistical potentials are used in the algorithms like Rosetta. So you don't really have a physical representation of the energy like we have usually we have. It's a kilocalories per mole, and in the Rosetta algorithm you have some kind of a Rosetta energy unit. So it's a kind of a knowledge-based function where they just did the literature parsing and devise the interaction accordingly. And they also provide in a lot of function within the Rosetta pipelines. Those who are interested in, you could just go to my publications where we're doing this analysis and trying to measure the stability of the complexes. So the next is the empirical function. These functions are based on the types of the interactions when you actually trying to analyze different descriptor properties and for your system, and then you have some kind of an empirical function. Then you devise them. Then you would probably improve this empirical function by adding more information taken from the literature and stuff like that. And the last one is the hybrid function, where you can use, actually the empirical function is also widely used or knowledge-based function is kind of widely used in machine learning techniques, because what you have in the machine learning, you have the information taken from the external database, or the empirical training of the system, of the model. So you do the training of the model, and you got the function which is related to this particular model. So you train this, and you got this kind of empirical function. And you can also use some kind of a knowledge-based information to train the model. So it's kind of a empirical / knowledge-based function. So you might come up with some kind of hybrid functions widely used in the machine learning techniques nowadays. And the combination of different functions, either it's empirical or knowledge-based. Probably you could also use the force field in a machine learning, but because the machine learning is lacking the physical representation of the binding affinity at some point, some very sophisticated algorithm, they're still lacking the physical representation. And this might be also the very a significant drawback of this technique. Okay, and the hybrid functions are also, by using the hybrid function, or by using, if you are using the force field functions, it's also possible to, like in Autodoc, you could also provide the physical representation of the molecules like this. You got Gibbs free energy, you got gas constant and room temperature, and the inhibition constant. And from this equation, you can calculate. You can actually calculate the inhibition constant. This is also very close to the probability. As you can see, the probability function also has the temperature, and actually, so this would be probably the most accurate function in terms of the comparison with the experimental data, because in the experimental data you have the inhibition constant, and you can actually compare this experimentally derived, the theoretically derived inhibition constant. And here's what if we are not successful with the molecular docking. You can see that we have a nice match here, and if the match is not good, then of course the energy will be very high. Okay, so, and I would like to say a couple of words about the new algorithm we have now. It's a diff-doc algorithm. It was published just a couple of months ago, and the guys from MIT using the kind of machine learning technique, epistemic uncertainties causes distortion and steric clashes. Yeah, and this is exactly what they have. They have the steric clashes like in the distortions. So it's also a big drawback of this algorithm, but still works quite good in terms of the clustering. And there are some other algorithms based on the regression, also the machine learning regression models, like equibind and tank-bind, but I didn't really test it, and the people from the MIT, they're claiming this is the best algorithm so far. Actually, it's a pretty long story, so we can try to go through this algorithm in the next lecture. So I think this would be more reasonable. Okay, so we will finish for today, and I will talk about it in the next